Hello dear students, welcome to the Medical Globe. Today's topic is polysaccharides from the chapter Carbohydrates. We have already discussed monosaccharides and disaccharides. Today polysaccharides will be discussed. Let's start with the definition. Polysaccharides, here if you see poly always means many, and saccharides mean saccharone, means sugar. So the overall meaning of polysaccharide is many sugar units. It means that in polysaccharides, one monosaccharide, if I write here M, you know very well monosaccharide, another monosaccharide, many monosaccharide when combined, remember that its number must be more than 10, must be more than 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and so on. I'm going to write dot, 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 and so on. Means in polysaccharides, there will be more than 10, actually in hundreds to thousands, units will be there. So this kind of carbohydrate will be called as polysaccharide. So here I'm going to write M. What is M? Monosaccharide. All the circles represent monosaccharides. Now, what are the example of polysaccharides in our daily life? The page that people use to write on is a very good example of polysaccharide, right? Because you can see here is cellulose are there. Plant cell wall contains cellulose. It is also a very good example of polysaccharide. And cotton is another example of polysaccharide. There are so many other examples of polysaccharide like starch, the one you eat the one you take from while eating potato or rice, these beans, these all contain polysaccharides. So one of the interesting topic is for today. Okay. Now the types of polysaccharide. This is more important to know. What are the types of polysaccharides? Polysaccharides are mainly divided into two categories. Number one is homopolysaccharides, and second one is the heteropolysaccharides. Today's lecture will be exclusively about homopolysaccharides but not heteropolysaccharide. However, we will give you some idea about the heteropolysaccharide as well. It will be discussed in the next lecture. What is a homopolysaccharide? If you see here, homo means same, and polysaccharides means units. Poly means many, saccharide means sugar units. So the overall meaning of the homopolysaccharide is that polysaccharide which contain same sugar units. Means all, if you see here is one sugar. If it is glucose, let's suppose it is glucose, second one also is glucose, third one also is glucose, fourth one, fifth one, 10th one, 100th one, 1000th, any number, all will be same. There will be glucose. Means in one polysaccharide, each unit will be same. Although they will be many in numbers, but they will be similar. All the units will be similar. Okay. Similarly, if one unit is fructose, the other unit is also fructose. All the units are made of fructose. So if all the units are fructose, this is again a homopolysaccharide because the units are similar. So in this case, when all the units are fructose, we will call it fructosan. When all the units are glucose, we call it glucosan. So glucosan, fructosan, these are a very good example of the homopolysaccharides. This was just to make you people understand about the definition of the homopolysaccharides. We have more examples of the homopolysaccharide that will be discussed today in this lecture. Okay, so what are the example of the homopolysaccharide? This was just a concept. Okay, here we will come here, have a we will give a slight touch to it, and then we'll come back. Hetero means different. If you see here, hetero means different. So polysaccharide means many units. Now if there are many units, let's suppose many units, this is one unit, this is another unit, this is third unit, that's fourth, and so on. If there are many units, if one unit is a glucose, other one is something else. Let's suppose this one is amino sugar. Let's suppose this one is amino sugar, and the second one is a sugar acid. So in this case, all the units are not the same. All the units are different. Means within one polysaccharide you will see different units. So when different unit comes, then this will be called as heteropolysaccharide, and that will be discussed in the next lecture. So now we'll come back to the homopolysaccharide. The examples of the homopolysaccharides are, number one, I will write it here in same place. Number one is starch. The first example is the starch. The second example is inulin. In starch we will discuss dextrin and dextran. These two will also be discussed. And then inulin. And third one is the glycogen. Fourth one is the cellulose. Fifth one is the chitin. So these are some well-known examples of the homopolysaccharide. We will start with starch. So first, second, third, fourth, and fifth. One by one, we'll discuss one by one. Okay, let's start with the starch. Let's start with the starch. Starch. A starch is a homopolysaccharide. Sentence number one. That is fine. Everyone now got this. That starch is a homopolysaccharide. The question is, the main idea is this: what are the units that combine to make starch? What are the smaller units? So the answer is, a starch is made of smaller units called D-glucose. So means a starch consists of many glucose units. Glucose, again glucose, again glucose, many glucose units when combined. Many glucose. I think once again there will be no need to write the actual structure of the glucose here, because it will consume our time, and we already know the structure of glucose in the topic of the monosaccharides. That's why I am avoiding to write the structure of glucose today in today's lecture. Okay, we have already discussed the bond between the two monosaccharides as well. So this is one glucose, the other glucose. In between the two glucose there is a bond. We call it glycosidic bond. Okay, similarly, all of these glucose are linked together by the help of the bond. We call it glycosidic bond, and that bond will be either alpha 1,4. That is, we should remember that what is the bond in the starch. We said alpha 1,4 is the bond, means from first glucose the carbon number 1, and from second glucose the carbon number 4 will be used, and the two units are alpha sugar. We know there are two types of sugar, one is alpha, one is beta sugar. So when both the two are alpha glucose, between the two alpha glucose there is a bond. The name of that bond is alpha 1,4. Alpha 1,4 means the carbon number 1 of the first glucose and carbon number 4 of the second glucose used. Similarly here again alpha 1,4, 1,4, 1,4, all of the units are linked together by the help of alpha 1,4 bond. And one thing more, if I suppose that there is a glucose unit, this is one glucose unit, and we can write here that this is carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, carbon number 5, and this one is carbon number 6. So remember that there is branching as well in starch. In starch there is some branching, means if you go like this, if you look at this, is called the main chain, the straight chain. Okay, so they call the main chain or the straight chain. At some point you will see there is a branch, there is a branch, more branches. This is one branch. Here you will find another branch. Here you will find another branch. So these branches are the characteristics of the polysaccharides, especially the starch and glycogen. They have so many branches. Now what we need to know about these branches, we should know the number of the bond, what number it carries by that bond. So just like here, the main bond was alpha 1,4, the main bond was alpha 1,4, and at the branching point, like just between here, the bond is alpha 1,6. That is the main point that we should know. How it is 1,6? If I continue this as like this, okay, this is going on and on the main chain. So here is carbon number 6. When another sugar comes, another sugar will make its bond here, exactly at this position, right? So its carbon number 1 will link with the carbon number 6 of this. So this sugar, this glucose has two bonds, three bonds. One bond with this sugar, second bond with this sugar, and third bond is with the chain one. So once a branch is established by the help of 1,6, then you will find so many units again linked together by the help of alpha 1,4. Again you will see alpha 1,4, alpha 1,4, alpha 1,4. Only at the branching point you will see alpha 1,6. So that was very important to know. That is very important to know, that we should know that number, which bond is there in starch. Okay, now we will come to the types of starch. If I write another thing, types of starch. Starch are of two types. One is called as amylose starch, and second one is called as amylopectin. Number one is amylose starch, and the second one is called as amylopectin starch. These are two types of starch. Now what we will do, what is the difference between these two? Here is the idea. Amylose starch is soluble, it is water soluble, and unbranched. There is no branch. Remember, there is no branch. As compared to amylose starch, amylopectin is not soluble and it is branched. branched. This is the main difference. It is branched. And if you look at the percentage, means if you take starch from somewhere, from any shop, from any distributor, if you take starch, then inside that starch you will find both amylose as well as amylopectin starch. So take a container, if you have a beaker, and in that beaker you have starch. So as a student may think that beaker contain either amylose starch or amylopectin starch. So the answer will be that beaker contain both the starch, amylose as well as amylopectin. There is a percentage. The percentage of amylose starch is 15 to 20%. This is 15 to 20%, and the second one is 80 to 85%. This one is 80 to 85%. Will be there. This is number one. Number two, it contains 200 to 1000 units. Amylose starch contain 200 to 1000 units. Means from 200 glucose units to 1000 glucose units you may find in amylose starch. Is here in amylopectin starch you will find how many units? Few thousands. It means in the book of Satyanarayan it is mentioned as few thousands. Means not less than 1000, more than 1000. So few thousands glucose units are joined together, and we know very well that in amylopectin starch there will be branching. Means this is the main axis, and you will find one branch here, you will find another branch here, you will find so many branches in amylopectin. Remember, at the branching point the bond is alpha 1,6, and this is also something that we should know. How many units are there in one branch? How many units are there in one branch? So this is from 20 to 30 units. 20 to 30 glucose units are in one branch. Means if this is one branch, this contains 20 to 30 glucose units. Every branch contains 20 to 30 glucose units, but overall amylopectin contains thousands of glucose units in that structure. Okay. Now, more interestingly, if we take a starch, a starch, here if I write starch and we hydrolyze it, let's suppose we just hydrolyze it, and for hydrolysis you have two options. Either you can use an enzyme amylase, or you can use any other acid, right? So if we hydrolyze the starch with the help of the amylase enzyme, then you will get many pieces or different lengths, and one of these pieces is, let's suppose this is one piece, and this is called as, okay, I will write so many pieces, right? So these are all the pieces that we got by the hydrolysis of starch. These pieces have intermediate lengths, different length. These are all collectively called as dextrin. We will discuss dextrin further as well, but remember, a dextrin, not dextran, because there is some issue. A-R-I-N, not R-A-N. Dextrin is nothing special, but it is the hydrolytic product of starch. So means if you hydrolyze starch, you will get dextrin. And that dextrin is little bit different from another thing, dextran, which will come later on. If you hydrolyze dextrin further, for more hydrolysis, you will get more pieces of shorter length. Means starch has long length, but dextrin small length. When you further hydrolyze, you will get more shorter pieces called as maltose. So yes, further hydrolyzed you will get maltose, and maltose we know very well, maltose contain How many units? Only 2 units. Maltose only 2 units. So you will get a lot of maltose disaccharide. And if you continue hydrolysis, you will get, means if you continue, you will get finally you will get many glucose units. Many glucose units. Lot of glucose units if you hydrolyze. Furthermore, you can't hydrolyze it more because glucose is the ultimate final unit, a smaller unit of the starch. Okay, and then inside the dextrin, some more points I also have for you people. If you see the color test, means with iodine, if we take iodine and mix it with starch, you will get some color, and that color will be the identification. At the lower classes, when we see that when you mix starch with iodine, you will get blue color. Yes, this is a kind of identification. If you don't know, if you have some unknown sample and you want to know either it is starch or glycogen or something else, so what you have to do, you have to just put the sample and mix it with iodine and then check the color. If the color gets blue, it is starch, and if red, it's called glycogen. Furthermore, in the dextrin, we got some new names for you people. That is, I have to erase it now. In the dextrin, here is dextrin. In the dextrin, if you see here, there are different types of name you will see. Number 1 is the soluble starch, and the second one is the called as amylodextrin. The third one is erethrodextrin, and last one is the erethrodextrin. Okay, the 4 different names are there. These 4 different names are actually, these are all related to the dextrins. These are actually dextrin, and you know very well that these are related to starch. So these are some sort of starch. Why we gave the different names? Because this is on the basis of the color. If you mix iodine with the dextrin and the color, if the color is blue, the blue color will be named as soluble starch, and if the color is violet, the name will be amylodextrin. And if the color is red, means maybe sometimes you get red color with it, it will not be glycogen, and then it will be called as erethrodextrin. And if there is no color at all with dextrin, then it will be called as erethrodextrin. So dear students, just the 4 names are there, and you don't worry about these 4 different names. Actually, these are dextrin, and dextrin is a hydrolytic product of the starch. There is one more different polysaccharide, in the name of which is slightly or more matching with the dextrin. What is that polysaccharide? This is actually the dextrin. Okay, here that is A and there is I. Dextrin is not dextrin. By pronunciation looks like these are similar things, but not. Dextrin is a polysaccharide, but we make it from sucrose. Means if you take sucrose, and in this sucrose you add a bacteria. There is some special bacteria called leuconostoc mesenterite. Here is the name of bacteria, leuconostoc mesenterite. Here is the name of the bacteria, and that bacteria will give us a new polysaccharide from that sucrose. Sucrose is just a disaccharide, so the bacteria will make a polysaccharide from this sucrose, and that polysaccharide is called as dextran. Of course, all the units will be again of glucose. Now let me compare the dextran with the dextrin. In dextrin and dextrin, there is a difference of branching as well. Means if we see in the dextrin, the first one is it is used as a plasma expander. Okay, right? The first one is we use it as a plasma expander. What is mean by this word plasma expander? Plasma expander means if during some emergency situation, during accident, during surgery, if your blood is lost and you need more blood, because less blood means less plasma and there is a chances of low blood pressure. So what we do, we introduce some fluid in the blood so that the volume of that blood is increased, but we cannot add any fluid. We add the solution of dextran. This is quite inert and it is injected, and now the plasma volume is increased, so the chances of hypovolemic shock is also decreased. So this is why we use a plasma expander. Second, we can also use the dextran in chromatography. Chromatography is also the uses of the dextran. And then we have one more thing here, the difference between dextrin and dextrin. In dextrin you can see there is branches, main 1613. Here you can see two types of bonds. One is called 16 and one is called as 13. These two types of bonds are there. Means 16 will be the main chain at main chain here. 16, 16, 16, 16, 16. But wherever there is branch, wherever you can see there is branch in the dextrin, at branching point that will be the 13. This makes a very good difference. We can differentiate dextrin from the dextrin. The first difference was that this is the product of starch and this is the product of bacterial action. Second, it is not there is no branches. Okay, no branch here, but here you can see there's a branch. Okay, and I think straight chain 14 and 16. Okay, dextrin. Here is 14 and 16. 16 means that there is branch. I'm sorry, I said that there is no branching. There is also branches, but the branching is at 16 and the main bond is 14. So 14, 16 in the dextrin, while 16 and 13 in the dextran. I hope you got this idea of dextrin. Now we will go towards next topic, that is inulin. Okay, so now it's time to get inulin. This homopolysaccharide is a little bit lengthy, so you have to keep patient. Inulin, and this is you can write it inulin. What is inulin? Inulin is also a homopolysaccharide, but you will see that there are many repeating units called as fructose. Fructose here, fructose, this is one unit, again fructose, again fructose. So when you see that the repeating units are, oh my god, repeating units are fructose, F, 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 F. So many fructose when combined together, many fructose, not glucose, then we will get a polysaccharide. Of course it's a homopolysaccharide because the units are same. So this kind of polysaccharide which is made by the repeating units of fructose, that is called as inulin. And inulin is medically very important, because if you want to check the function of the kidney, either the kidney is functioning well or not, then you can use this inulin to analyze the function of the kidney by the help of a test called GFR, glomerular filtration rate. Glomerular filtration rate. Then we come to the third one, that is glycogen, and last we will go to the cellulose. So the third one is now glycogen. Glycogen is another homopolysaccharide. Again, all the units are glucose, glucose, glucose. Many glucose, when linked together we get glycogen. But what is the difference of glycogen with starch? Starch also contains glucose units, glycogen also contains glucose units. There are two main differences. One, starch is the reserve food in plant, but glycogen is the reserve food, stored carbohydrate in animals. So this is the main difference. Starch for plant and glycogen for animals. Even some fungi also contain glycogen. So glycogen has many glucose units, and one more thing, difference between starch and glycogen. Starch contain branches, yes. Glycogen also contain branches, but very the branches are so many as compared to starch. So here, if I write here, this is one chain, one branch, another branch, another branch, branch and branch, branch and branch. So many branches are there. This is another difference of glycogen with starch. Means starch contain branches, glycogen also. Starch less branches, glycogen many branches. And one thing more, when glycogen is synthesized in our body, especially in the liver and the muscles, right? Whenever its synthesis start, our body needs a protein to get start the synthesis of this glycogen. And at the beginning we need a protein here. This protein which will help us to synthesize the glycogen is called as glycogenin. This is called as glycogen. Glycogenin is a protein that will help us. It is a kind of starter to start the process of synthesis of the glycogen. Okay, nothing special was there. Okay, if you take iodine, mix it with glycogen, you will get red color. This is identification test for the glycogen. Nothing special here now. Okay, there is a azan, so we will continue in my next lecture. Thank you.